And greetings. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova. It's a pleasure being your host tonight. This is going to be a brutal show. I'm trying to forewarn the readers, but I think it's a chance for the viewers to see what life as a professional researcher in creationism is like. I hope you can hear me. There's someone I haven't seen. Mitchell, well, greetings. So I'm expecting to see Dr. Dan in a little bit, and I just have to, since I don't have the co-host to help me with the controls, I have to operate this myself, so it gets to be a little bit clunky. And Wesley Coleman tells me the audio is good. So let me just get some announcements out of the way. First off, there is a party happening at Logical Plausible Probables channel that starts at 10:30. If any of you are party animals, you can wander over there. I don't take any offense. I actually would want to spend time over there, but I just figured this would be, I have to keep moving on and keep trying to research. So hang on, let me get my audio adjusted. And I'm also going to get some things set up here. Give me a moment. It's always tough when I don't have a co-host. The co-host could handle a lot of this for me. And when I have to run the show solo, it's a little harder. So some announcements. I dropped it in earlier in the side chat that there is going to be a webinar. I'll show it here. There will be a webinar at the Discovery Institute, and I dropped it's going to be around, I think, 11 a.m. Pacific time, or 1 p.m. Let me just check the date and time. I don't want to get anyone messed up here. I'm going to confirm it. It is 10, I'm sorry, it's 10 a.m. Pacific time, and that would be 1300 Eastern time. And they're going to be releasing a new animation of the topoisomerase enzyme, which I've talked about for the last 2 years on the internet. And it's been, this animation has been about 1.5 years in the making, and I've been waiting, and I've been bugging Joe, when's it going to come out? And he said, it's coming, it's coming. So now it's finally going to be released. And so we're all excited about that. So that's a little announcement. So it's free. You can watch, and you can see the you'll be able to see the premiere of this. So let me switch something here. StreamYard and PowerPoint don't get along well. So let me see if I could reset this. Nope. It put me on the last slide. So let me just show a little bit. So Joe DeWeese is a young Earth creationist. Joe DeWeese is a young Earth creationist, but he's published in the top tier scientific journals. So he's very accomplished. He has just a tremendous resume, curriculum vitae. I had the privilege of having some publications with him on the topoisomerase, that he was my mentor. Dr. John Sanford sent me off to study with, to be mentored by Dr. DeWeese on some of these projects. This is, that graph was actually developed by yours truly. So that was 2019. This is a publication in 2021 in the FASEB journal. And there we are again. But it was fun to be co-author with him and the Creation Research Society. And we were talking about topoisomerases and a little bit less encumbered because we were freer to speak our mind. But we really took most of Joe's secular research and a little bit of mine, and then just added some considerations about why we don't believe it could evolve naturally. So I hope you enjoy. Some of you are able to go to the seminar. I hope that they will release recordings of it. I think it should be fun. And more importantly, I expect they're going to have just a killer animation. I can already see that it's preparing to be a much better than any of the other topoisomerase videos. And because topoisomerase is so important to chemotherapies, this is really a great way to introduce intelligent design, because when people are Googling for videos in their universities to show topoisomerase, they're going to end up in the Discovery Institute website. So this is brilliant, really, on the part of the Discovery Institute. And, you know, God put a young Earth creationist at the forefront of this. He's also, Joe is one of the special young Earth creationists who's also part of the Discovery Institute. He's a junior fellow there. Now, Dr. Dan has said he'll join when he either gets it working or gives up. So good luck, Dr. Dan. So tonight's topic is going to be something I learned about at the Institute of Creation Research when I visited Dallas on January 21st. Dr. Dan is an evolutionary biologist. He has obviously a very different view on this. I'm not, don't expect for this to be a debate. We're probably just going to open the show if he's able to join, and we're just going to disagree and say we have come to different conclusions as to the meaning of the paper. There's some things in the paper I would obviously disagree with, and I'm not trying to imply that the paper is telling anyone that young Earth creationism is true. But there are some signatures in this that are of interest that if the paper were just maybe looked at from a little different perspective, and we consider the numbers, and especially if we're able to duplicate this in many, many animal species. If you get the same signature that many of the disease-associated mutations, the proper term in this paper is single nucleotide variants. If they all emerged in the last 5 to 10,000 years, not all, but let's say the overwhelming majority, say 73%, 75%, emerged in the last 5 to 10,000 years, that would raise an interesting problem. Why would that be across so many animal species, like say mammalians? That would be a testable prediction for a speculation. So at this point, I'm going to just throw it out there as a speculation. We could probably do this, not that anyone has the money or incentive at this point to do that for all the mammalian species, the extant mammalian species. But what if we get that same signature hypothetically? If we get that same signature across mammalian species, that would be good evidence of, say, the flood. Now, Dr. Dan earlier pointed out that, well, the study said it goes back to 80,000 years. And again, that would be one data point that the YECs would have to wrestle with and say, okay, that is what the paper says, and that disagrees with young Earth creationism. How can we remedy that? So, you know, how can we explain that? And that'll be something to deal with. But when I saw this right away, I said, I want to know what it's about. So also, this is also, I have a policy of trying to prepare young Earth creationists who may be going into a university environment. So if they're going into biology curriculum, they're almost 100% likely, if they're at a secular school, going to be studying biology from someone who believes in evolution. And they're going to be in an environment where a lot of their ideas and beliefs are challenged. Someone like Joe DeWeese was able to get through the program as a creationist, as a young Earth creationist. He got a he got his PhD at Vanderbilt University, and he became an adjunct assistant professor at Vanderbilt University School of Medicine, and then an associate professor at Lipscomb, and now he's a professor at Freed-Hardeman, his undergrad alma mater. So it's certainly possible for a young Earth creationist to start that way, go to school and stay a young Earth creationist. In contrast, myself, I was a theistic evolutionist in high school. Just before I graduated, I became an old Earth creationist. I rejected evolutionary theory and then went off to university. Was an old Earth creationist for most of my life up until about 2000, had a crisis of faith, explored origins, and that restored me to faith. And I also ended up getting closer to being a young Earth creationist. And praise the Lord, Dr. Dan is here.
Hey, can you hear me okay?
Oh, I could hear you. Wonderful.
Cool.
I'm going to remove my duplicate. Dr. Dan, let me introduce you to my audience. There was an informal poll of creationist biology graduate students, and they were asked, who would you like to have as your professor of biology, Dr. Dan or Kent Hovind? And Dr. Dan was unanimously voted as the man. So I don't know if you heard my intro, Dan. I was saying that you know, we're going to disagree about the conclusions and the interpretation of the paper, and it's only fair to, and it's only fair to point out that the paper is not endorsing young Earth creationism. It has some dates that we wouldn't agree with, but maybe we could just leave the disagreements off the table for a little bit, because
let's treat this, let's go through the paper and see what they did and what they concluded and why, because it's cool paper.
Yeah, this would be a good I'm trying to also just give the viewers kind of an idea what it would be like for a creationist biology student studying under an evolutionary biologist. And it's feasible. It can be, depending on the professor. It just, a lot depends. Even if it's an evolutionist student with an evolutionist professor, there are always these dynamics that you just don't know how it's going to turn out. But I'm just saying, this is a little bit of what it's like for a creationist to study. Has to try to sit there and be tutored and taught and learned. So there will be my colleague Rebecca Davis is having Dr. Dan on her channel, Bread of Life, and the title of that is Evolutionary Biologist Schools the Creationist. And actually, Dr. Dan's going to have a chance to do that twice, because he's going to school me today. And
I don't know about that. We'll see.
But I wanted to say, my boss, Dr. John Sanford, sent me off to the NIH to learn from people like Dr. Dan. And I wasn't trying to be patronizing, brother. I was just.
No, I laugh at the thing with Rebecca on Friday. It's it's the thumbnail is like, oh, it's going to be all adversarial. And it's like, we're just going to chat about evolution for a bit. It's not going to be like, no one's throwing bombs over here.
Now, I got to tell you, I looked. If people were wondering about the thumbnail and I was showing Dirichlet, I clicked on one of the reference papers and I was just browsing it. And I saw this. It said, using the overlapping genotypes of the Sanger ENCODE and SNP chip HapMap projects to train a Dirichlet mixture. I'm just like, oh my goodness, I'm toast.
I don't know what that is. It doesn't matter.
Okay, Dirichlet, I learned in electrical engineering, and I'm just like, where did this come from? What's this
I've
doing in
never
the
heard.
biology?
I've never heard that before in my life. I don't know what that is.
Okay,
I could
so
talk about the other stuff in this paper. I went through and I highlighted it, and there's like, I get what they did, I get what they found, but like that part, I, I got nothing.
well, this is actually of interest to Emery and I, how they're able to reconstruct, calculate. I've been saying I wanted to know how to calculate a most recent common ancestor of an allele in an expanding population. And it's just really hard to just, I have Felsenstein's graduate book, and I'm just like, it's still kind of hard to figure out what formula we throw. So with that, why don't we, is it okay if I just post the paper and we just kind of read?
Yeah, just put it up. I got
Okay.
it right
Now
here. I've got
I
mine is
really
just
actually have
yellow.
I have tons of biology questions for you, actually. So
Let's
I have to
do it.
make a confession to make. I like proteins. I actually don't like genetics. I only did it because
Breaking
I
my
was.
heart, Sal.
I'm
Breaking my
sorry.
heart.
I
It's
broke Dr. Sanford's heart too. So
I mean, if you're going to talk evolution, it's all genetics. It's all about the genetics. This is where it's at right here, because this paper is such a great illustration of some really important evolutionary concepts. So we'll get into, what they found and everything. And it's like reading it, I'm going like, okay, that makes sense. All right, I like this. Oh, this gets into the my thing in graduate school. A large part of it anyway was codon bias and synonymous versus non-synonymous codons, and that get into different types of substitutions in here. I like this a lot. This will be fun.
so I'll try to be cognizant that we do have an audience here. Some of them are sophisticated, and
Yeah,
if we might have to. I hope you don't mind if we try to explain terms, and actually it's good for me to review.
yeah. No, you should always do that.
All right, so I'm just going to read here. Establishing the age of each mutation segregating in contemporary human populations is important to fully understand our evolutionary history and will help facilitate the development of, well, why don't I read the title? I'm sorry. Oh,
yeah, let's start there.
I need to explain something. There is a tradition, what they call journal clubbing, where scientists, researchers get together and review journals and they talk about it. It's maybe you could explain why they do that, Dr. Dan.
I mean, it's a lot of just like graduate seminars in grad school. It's a journal club, right? So when I was in graduate school, the ecology and evolution department, we had our evolution journal club every semester, and everyone that participated, you'd kind of have 8 or 10 or 15 people, and kind of each week somebody else would be like, here's a thing, and then you very briefly present the paper, just 10 maybe 15 minutes, and then everyone had gotten it, the week. Normally it's like the week before you'd be like, we're doing this paper next week, so you have a week to read the paper and kind of go through it. And then you talk about it. What do you think of the techniques? Were they appropriate for the question? Were the conclusions adequately supported? Was there anything that the authors failed to consider or should have done differently? And it's a really great way to get out of your bubble, right? Because every scientist, and this is true for people who, have been, faculty or research scientists for decades, and it's true for graduate students, you get tunnel vision, right? You get into your thing that you work on day in and day out. And outside of that, you kind of don't have a good feel for what's going on, even if there's other research going on literally next door to the lab where you're working. If they're working on a different system, then I have no idea what they're doing, right? Like I was working on viral evolution in grad school, and it was in a building where you also had like the plant science department. So you had like a lot of like plant biotechnology stuff going on. So like the lab adjacent to us, like I had no idea what they were doing. I don't know anything about like the plant system they were working on, right? But you get people together from all these different backgrounds into the same room to talk about the same thing. You've got a bunch of people coming at it from different angles. So you, as protein science, biochemistry kind of angle, are going to approach this paper very differently from me, an evolutionary genetics angle, right? And you're going to get very different things out of it that jump out as like the important conclusions. And the other great thing about that is you're going to come in with different levels of understanding of the techniques that they used. So somebody in the room is going to be able to do like a really good job, hopefully, evaluating those techniques, right? Whereas for most of the people in the room, it's not necessarily going to be techniques that they are familiar with. And that's okay. The general rule when you get a paper for the first time, and this is just like a first pass, like bare bones kind of do I basically get it or basically not rule, is title, abstract, and then the figures and the figure legends. And if it's a well-constructed paper, you could pretty much get the gist just from that. And that's literally what I did today. Earlier today, I read the abstract and went through the figures. And Sal, that's what I commented on in the chat for this video earlier today. And then since then and now, I've gone through and I've read the paper and I've highlighted. So I've actually like gone through the text itself.
I didn't know that.
That's kind of the approach is my first pass. I learned this when I was in grad school. It's just like a general kind of good practice is first pass, just abstract and figures and get the big picture and then go through and get the nuts and bolts. And the reason for that is it's really hard to come into a paper cold, especially if it's not your specific specialty, and understand kind of the gist of the case the authors are trying to make while you're also trying to like wrap your head around the techniques and the methods at the same time. So you kind of separate those into separate readings, and that makes it easier to grasp what's going on.
Actually, I didn't know about that looking at the figures. I usually go title, abstract, and conclusion. But I will tell you, even though it's tedious, I can't say that I ever regretted reading any paper. There's always something to like, oh, especially actually, ironically, the ones that I had no clue about. I'm just like, I didn't even know that discipline existed, or I didn't know that term. Let me Google that, and that's an interesting term. So there's just really not enough hours in the day, and there are people. I've heard of some really excellent scholars who recognize, I can't believe how many papers they've read. I mean, they will dig. It's just...
I will tell you, though, you hear people who will say, you know, I read X number of papers a day or X number of papers a week. Reading it as though you're reading a novel is not really reading a paper. You really got to dig into it, and you're sitting there, you know, you're Googling terms, you're cross-referencing it, you're like, they reference something, like in this one, you see that, oh, we did XYZ technique, as in, and they reference another paper. So if you're like really reading the paper to understand, well, now you got to go chase down that reference and see what their methods were, right? So to like really read a paper in a comprehensive way, it's an afternoon project. It's like, okay, I've cleared my desk and I'm going to do this paper this afternoon, right? And I'm going to take 3 hours and do it. It's you know, this one, when you, I downloaded it as a PDF, and the text is really only about 4 pages. It's a half page, 3 full pages, and then another half page. And that's still going to take you easily, like, if I'm doing this like really comprehensively, it's going to take me an hour to go through those 4 pages. It's not like sitting and reading a book.
So again, this is just, thank you for that information. And this is why we call it Journal Club. And I call it journal clubbing, or nerds go clubbing instead of night clubbing. So let's look at the, the title of the paper is Analysis of 6,515 Exomes Reveals a Recent Origin of Most Human Protein Coding Variants. So for the way I read this, just the title, exomes is the mRNA transcript, or just kind of like the exons concatenated together to make a protein.
Mm-hmm. Yeah, the stuff that gets transcribed. Yeah.
Okay. And being a protein guy, I don't like dealing with non-coding DNA. So this is important, that we're not dealing, this paper's not dealing with non-coding DNA.
They do mention it in a few places, actually. They focus on the coding stuff, but in a couple places they do mention, let me go down to the figure here. It's, here we go. Yeah, they do have non-coding kind of as like a control region, basically. So they're looking at we'll get into it, but they're looking at like how long, when a mutation occurs, how long does it persist in the population, and like the baseline for non-coding, with small exceptions, is that it's going to be not sequence constrained. So the a new variant appears and it's going to fluctuate primarily due to drift rather than selection.
Let me acknowledge, Wesley, you're very, very generous. God bless you. So we're going to try to deliver. We're going to give you as much of a free college education here as you can get, graduate level too. I will point out there are 15 authors. What everyone has noticed is every paper now is starting to have multiple authors. The laboratories and projects and teams just get bigger and bigger. It's just getting insane. Just needed to point that out. So acknowledgement to all the authors there. So just start reading it. And I may pause to ask you a question. Establishing the age of each mutation segregating in contemporary human populations is important to fully understand our evolutionary history and will help facilitate the development of new approaches for disease gene discovery. Okay, I'll just pause. That means, I guess, they're going to try to find out what genes are associated with what disease.
Right, because you can survey if you know, I read this as if you know like the distribution in terms of the frequency, or you could do this kind of analysis based on population level surveys and work backwards and figure out the approximate age of a variant, and you know that disease causing variants tend to be young, you can focus your search instead of having to do like full genome, like actual like genetics analysis. You can focus your search based on the age of specific variants, because you know if a variant has persisted for, to look at some of the data in here, maybe 20 or 30,000 years, then it's likely that that variant is not associated with a disease, because if it was, it wouldn't have persisted for that long. So this allows you to focus your search space in terms of what specific alleles may be associated with diseases by basically saying, if it's older than X, we can eliminate that because it's highly unlikely to be disease associated.
I wouldn't have interpreted it that way, but we'll move on.
Let's keep going, because, well, how did you interpret it? Because my interpretation is not the only one, and it
okay. Well, maybe we'll discover. I mean, it's intuitive to think that a really severe. It is correct that a really severe variant that is a deadly mutation, it's going to select out of the population. However, in an expanding population where you can get kind of some isolated pockets, there is kind of a box when you have small population sizes that are not well stirred. You can get a diseased population that can persist. But why don't we keep going on? And just find out, because we're only at the third sentence. I don't want to get
let's go on
speculation. Large-scale surveys of human genetic variation have reported signatures of recent explosive population growth. This is one reason I was really interested, because I like exponentially growing populations. It makes the math easier. Okay. So I already liked it from that. Notable for an excess of rare genetic variants, qualitatively suggesting that many mutations arose recently. To more quantitatively assess the distribution of mutation ages, we resequenced 15,336 genes in 6,515 individuals of European N equals 298. So N, that's the number of people. I guess.
The number of Yes. So that's, let's see. Yeah. So that's going to be the number of people, right? Right. So it adds up to the 6515. Yeah.
Okay. Thank you.
Like, let me do math real quick here.
Well, and I only learned that N is significant. By the way, I don't have actually, my statistics background is in gambling. Okay. So they have these, if they have stuff with p-values or N, I'm just like, okay, I'll probably have to count on you. So American ancestry and inferred age of 1 146,401 autosomal single nucleotide variants. Okay, autosomal that would be from a chromosome that's not sex determining?
Yeah, so it's not the X or Y chromosome.
Okay.
So we're excluding X and Y chromosome, and the reason for that is that the X and Y chromosomes are, in general, non-recombining with each other, that is. There's a small region of the X and Y that'll recombine, but other than that, they don't recombine with each other. Obviously, XX karyotypes will recombine, but it makes the math trickier if you have to account for that. So it's easier to just be like, just don't include them.
Autosomal. Okay. Now, single nucleotide variant means you have basically one base or base
just
pair
a mutation.
changed.
Yeah.
Okay. Mutation.
And for everyone watching, for both our viewers, if you anytime you see SNV for this context, it's just a fancy way to say mutation. So we're just talking about single base mutations here.
Yeah, because there are varieties of mutations like insertions, deletions, frame shifts.
Right. They can be regions of varying length. Right. But for today, we're just saying, an A switches to a G. Right. That's an SNV.
Now I've seen the term polymorphism. Is there?
Yeah. It's
same thing.
Same basic thing. Yeah. I mean, polymorphism, I guess, would be a little. You tend to think of like polymorphism at the population level. So like within a population, if you look at a site and some of them are A and some of them are T, you say, okay, that site is polymorphic within this population.
Okay, thank you. We estimate that 73% of all protein coding SNVs and 86% of SNVs predicted to be deleterious arose in the past 5,000 to 10,000 years. The average age of deleterious SNVs varied significantly across molecular pathways, and disease genes continued a significantly higher proportion of recently arisen deleterious SNVs compared to other genes.
Oh,
disease genes.
One second, Sal. I'm listening, but I'll be right back.
Okay. So Dr. Dan's still listening, but I'll just keep reading. And disease genes contained a significantly higher proportion of recently arisen deleterious SNVs compared to other genes. So other genes, I presume, are not diseased. Furthermore, European Americans had an excess of deleterious variants in essential and Mendelian disease genes compared to African Americans. So when Dr. Dan gets back, I'll have to ask him what Mendelian disease genes are. And also what essential, I think, is, what would be considered essential disease genes. Consistent with weaker purifying selection due to the out of Africa dispersal. Our results better delimit the historical details of human protein coding variation, illustrate the profound effect recent human history has had on the burden of deleterious SNVs, segregating in contemporary populations, and provides important practical information that can be used to prioritize variants in disease gene discovery. And then when Dr. Dan gets back, we can just survey the figures.
I'm back, sorry.
Oh, great. So why this ended up on this channel was that number of 5,000 to 10,000 years will get the attention of a young Earth creationist real fast. So we're going to skip that discussion for now, but just so you know why it was of interest. And this was actually easier for me to comprehend than Nathaniel Jeanson's work, so I was actually liking it for that reason. So there was a question: what are Mendelian disease?
Okay. Yeah, so they
Mendelian
break it into a couple of categories, right? They say there's Mendelian and then there's essential, right? So the Mendelian refers to diseases, I interpret that as recessive, basically. So you can have homozygous dominant, where you have two of the non-disease alleles, be heterozygous, which would be a carrier for that condition. So you have one functional allele and one disease-associated allele.
Okay.
That's a carrier. And then homozygous recessive would be having two disease-causing alleles, and then that means you're affected by whatever the condition is. So it's hard to completely get rid of Mendelian traits like that because there's no purifying selection acting on the heterozygotes. You're not selecting against the heterozygote, so the recessive allele persists within the population. Does that make sense? That's the.
Oh, Dr. Dan, there's we're getting someone put something there, trying to get rid of it. Anyway, sorry, I have to deal with the side chat.
Should I delete somebody? Who should I delete? Oh, yeah, I got him.
I got him. Thank you. So
there you go.
I don't know what the purpose of that is, except to just vandalize. I mean, nothing's accomplished.
All right.
As part of the, okay, so we can go through. I mean, that's really nice. So we can now go through the figures.
Yes, let's go down to the figures, because the figures really tell the whole story of what's going on here. The paper, it's fine. It's a litany of, we found this, we found this, we found this, but it's all in the figures. So we could look at the figures, and this really tells the story here. So let's see what we have. Let me pull up figure legends here. All right, let's look at figure 1. Okay, site frequency spectrum. Okay, so figure 1A is, let's see. So basically, how frequent are the proportions? So basically, what this is showing is that most of these variants are either very rare, right? So on the far left side, it goes up. That's basically saying the frequency is extremely low, right? And then it drops off very quickly. So very few are more frequent than 0.02, right? It doesn't, between 0 and 0.1, there's a giant drop off very quickly, right? Like right below where you have your cursor right now. And then
Dr. Dan, I'm sorry to interrupt you. There's a lot of staticky sound when you talk. Is that your mic? How did you know that how did you know you're having some problems with your mic?
Oh, well, the mic I normally use wasn't working. So I'm on a different mic. Is it static?
All right.
Hang on a second. Hang on one second. Is that better? Is that any better?
Unfortunately, no.
Hang on. You know what? Let's see if it changed my source. Hang on a second. Man, how's that? Is that?
Yeah, that's better. What'd you do?
I changed the source and then I changed it back.
Oh, it's really good. What do you think, guys? I can hear him better.
Yeah.
All right. So
about that.
I'm a little dense here, and this is where.
So this
now
is,
you're going to see how I crawl through. I can't even understand the graph, honestly.
I'll tell you, this is not critical. This is like not super important for this paper, but basically this is just showing the distribution of the frequencies of these. So if you look at the y-axis, and by the way, this is just a convention for everybody, when you're going through a figure, start with the axes, then get to the data, right? So on the left side, we've got our y-axis, and that's just it's a log scale for the proportion. And then across the x-axis is frequency from 0 to 1, right? So if your frequency is 0.1, that means a particular mutation or a particular variant is found in 10% of these sites within the population, right? If your frequency is 0.6, then it's found at 60%. So what we can see here is that the vast majority of these variants are extremely rare, right? That's all the way up on the left side. And then it's a log scale. So you go up on the left, and most of them are found in almost 0% of the population. And then by the time you get to 10%, it's look at where it is on that log scale. It's like 0.01,
Okay.
right? So
right.
it's most variants are extremely rare. Like that's that.
No, I need to just apologize to the viewers. When I expand it so you could see it, it chops off the other graph. But
you could do one panel at a time. That's okay. I think the big one really is it's
sorry.
You know, I really think part C and D are
I just Dr. Dan, I just blew it up. Sorry guys.
That's okay.
Okay, so I learned something. This is hard to do on the net when you're trying to get figures so you could see it. Let me try to expand a little bit more.
It might work better if you get the PDF and share the PDF instead of a browser window, because then it's easier to zoom.
Dr. Dan, great. You have some great ideas today.
I got a lot of practice doing Zoom school over the last couple of years, so a lot of practice.
Before I forget, I may have asked you this, and I keep forgetting who I may have asked. Do you have any? I have a tablet to write. Do you have one of those so that you could highlight?
Yes, I can. Yeah, I've done that. Yeah, it's good. I like doing that a lot.
What do you use?
I just use an iPad.
Okay. All right. So how do I get rid of this panel? Well, let me just try to download this and then open it. Maybe it'll let me open. I'll put it down into my documents. Hopefully it'll let me open it. I can't get rid of this other window here.
I can share it too if you want. The only difference from mine is I've like barfed yellow highlighting all over mine while I was reading it. But other than that, it's the same.
Do you want to try?
Yeah, let me try.
That's great.
Yeah, because my computer's being, we'll see. It's misbehaving tonight, but we'll see. Boom. Let's share. Okay. I want to do this. I think that's right. Preview. Yes. Allow. The browser can't access your screen. Hang on. Let me try again. Hang on. Probably just did something wrong here. Hang on a second. Block. No, don't block anything. Why would you block? I don't want you to block it. I want to share it. Allow StreamYard to see your screen. Opera. No. Preview. Yes. Allow. Let me leave and then come back. I'm going to try to share it.
Okay.
Let me leave. I think I messed up a permission at some point because I was futzing around with settings before when I was trying to get the mic working. So let me leave and then come back and I'll see.
Okay.
Sorry. What a cluster. This is bad at things tonight. I'll be right back, everybody.
Alrighty. So I hope Dr. Dan's mic still works when he comes back. So he might be able to share his screen, but Dr. Dan got to come back. So, all right, guys, this is academia at its best. You're seeing what life is like as an academic. So in the meantime, I'm going to try to go back to sharing my screen. And see if I can get that graph going, just in case Dr. Dan is. And there, they made the other figures. And see if I could. Oh, Dr. Dan was right. We could zoom. Oh. All right, you can try.
I got mine shared if you want to look at that. There we go. So now what we can do is we can just zoom in on this. Okay. So this right here, this is what we just did. Basically, you've got your proportion of your variance right here from 0 to 100 on a log scale. And down here, we've got frequency from 0 to 1. Basically, this peak right here just tells you that the vast majority, anything basically over a frequency of, point or below a frequency. So the proportion, what is that, 0.1? So let's 0.1, 1, and then 10%. Like every variant is rare, right? Because you look at its frequency right here, there's like nothing. Once you get to like 5%, they're all super rare, right? So then you go across here, and then you get some that are enriched, where it's almost everybody has a variant. But in the middle, like that's 0.01%, right? Almost everybody's below that line. So these variants are rare, is the idea there.
Oh, Dr. Dan.
Not breaking.
Wow, this graph just blew my mind.
I mean, this is something that we've all heard before, that everybody carries a bunch of unique mutations, right? So the fact that there's 3 billion bases in a haploid human genome, 7 billion people, if you survey people, most of those mutations that any individual has are going to be relatively uncommon, right? There's very unlikely that the same mutations land in the same places across lots of other people, right? So that's basically what this is showing.
Yeah, it does surprise me that we have that kind of U shape, that it's not kind of flattening out toward the end.
Yeah, you get this, right? I think what
those
that
must be the common alleles.
So those are the very common variants, right? So, well, think about it. If you have something that's very rare, if you have something that's present in 1% of people, right? You necessarily have some other variant that's present in 99%.
Yeah.
Right? So you're going to have a little bit of symmetry to this. Now, it's not going to be perfect because you could have an A, a T, and a C at the same site. And maybe the T and the C are very rare, but then the A is more common, right? So it's not going to be perfectly symmetrical, but that makes sense.
So actually the ones then that are on the left-hand side, DAF again stands for, and I didn't read the legend here. DAF.
Something frequency. I think it's something frequency. You have to go up in the paper to find it. It's something frequency.
Okay.
Here, let's see.
A DAF.
Well, that's wrong. Yeah, I don't know. I don't remember from reading it. Something frequency. Okay.
Let me.
So, I can't. Let's see.
It won't let me search on it.
It's actually not in there. It didn't pop up, but maybe it's a problem with the PD searchable. I don't think that's an issue, but it didn't pop up for me. Actually, let's see if I can find something. It's definitely in there. Yeah, it's searchable, so it's just didn't have it. So I'm not sure what that is. It's something frequency. I'm
sorry to be anal. That would just make me feel better
fine.
if I knew what the acronym was. And
site frequency spectrum, but yeah. So that's the proportion, and that's the frequency. Something, yeah, I don't know. The F is frequency. I can give you that much. By the way, in the chat, just let me know if the sound is okay.
Okay, I'm sorry to ding the paper because it's a Nature paper, but that would have been helpful if they if they made it easier to understand what DAF. So Lord willing, we'll figure out what that is.
The key is the frequency. That's the key thing. Now, you hop over to panel B over here. This isn't that groundbreaking. I mean, it's when we talk about like young Earth and old Earth stuff, like this is relevant because it's telling you the age of the mutations, and this is a weirdly done graph. So this y-axis right here is relevant to this x-axis, and then you have these two curves. But then inset from here, if you're just looking at like the first 50,000 years, so you've got 200,000 years right here. If you just zoom into the first 50,000, this is what it looks like, where you've got age in years for the variant and the proportion of variance. So what you see here is, right, it starts at 40, right? So this 40 and this 40 are the same, and it goes up to about 90. So it's saying within this 50,000 year range, 80 to 90% of your variants originated within that range. Now, it's not saying the genes originated within that range, but it's saying that the variants that are present within human populations currently today originated in that range.
EA stands for, is that another
European American and African American.
African American.
Yeah,
Oh,
and
that's
the reason,
okay.
and the reason you have more variation among African American populations is that these are more admixture within the population and less genetic drift, because they are historically larger populations with fewer bottleneck events. Whereas with the out of Africa migrations, you've gone through a bunch of bottlenecks, so you lose diversity and you lose population size, so you have more drift in the European populations, which leads to this this distribution where more variants are recent. So you cumulatively have more of the variation at 10, 20, 30,000 years compared to the African-American populations, just because you've had less drift within the African-American populations.
Okay,
so this is again, this is like setting the stage. This is not like the super big finding. It's exactly what you'd expect, basically.
Thank you.
So then
I'm feeling already better that I'm getting this. I'm comprehending this.
And then this figure, I'll tell you, Sal, this figure is not super groundbreaking until I think D is really cool. I'm excited to get to D, but this part is also exactly what you'd expect. So what we have here is we're looking at the age within the two groups for things. This is like the overall, right? So the overall averages are like 25,000 years and like 30,000 years, or I guess like 50, that like 35 and like 50, give or take, right? And then, but for the ones that are shared between the two, now they're going to be older. Well, that makes sense, because that has to be before they split historically. So that has to be before the out of Africa migrations. Like, that makes perfect sense. That would be older. And the ones that are specific to each group are going to be younger. That makes perfect sense. That doesn't differ at all from what we'd expect. So, not groundbreaking.
Okay.
But the next one is where it gets cool, because the next one is exactly what we'd expect. But like, I think it's relevant for what we're going to talk about. Should we hop over to D?
Yeah. Go ahead.
All right. So this breaks it down by where. Oh, I actually have to zoom out a little bit to fit this whole thing. Okay, here you go. So this breaks it down by the type of variant. So like, where is it in the sequence and what does it do? So you've got on the y-axis, you've got average mutation age, KYR, K for thousands is 1,000 years. And then you've got splice sites. Sal, I know you're a fan of spliceosomes. So if you mess with a splice site, that gene is not going to work, right? So you have your,
bringing back some memories here.
We've talked about this at some length.
And what do they mean by splice site? Is it like the AG or whatever,
introns.
where the intron meets the? I mean, that's a really specific. That's a very specific location.
Yeah. So if you have a mutation at a splice site, that might cause a problem.
You could lose an exon or more.
Yeah, and then that protein's not going to work. So what this is showing is that
the reason I smiled was I said it brought back. There's that interferon lambda 3/4 pseudogene that I studied in school. And
nice. Oh, see, I thought you were talking about the time we spent like a week on Reddit talking about like type 2 introns and bacteria.
I've since forgotten that.
Oh yeah, we're talking about like self-splicing introns and stuff. It was fun.
Yeah.
We had fun. We had a friend who doesn't know. Sal and I met on Reddit. We've had fun on Reddit over the years.
Yes. So we have the splice sites are recent.
So basically, if you have so the splice sites were on average the most recent variants that we find. The variants in the splice sites, they tend to be limited within the population, indicative of a relatively recent origin for those variants. And that makes perfect sense, because if you mess with a splice site, you're very likely to break that protein. And then if you break the protein, you're selected against, because now you've inactivated a gene or something, right? And that could be a problem. So
go on.
So it makes sense that variants that occur within splice sites are going to be rare, because if you mess with them, they're going to be selected against fairly strongly.
What's the politically correct term? I mean, do we say ethnicity for African American or European? I mean, I think race might be the wrong.
I mean, races are made up. You can for this one, you can just look at your study groups as two groups.
Okay, thank you. It's amazing how it differs between the study group, the African American versus the European. You really do see a different signature, which indicates different population structure and history over time.
Right. In African populations, you haven't had a series of bottleneck events. And anytime or I should say founder events, right, where you have a large population and then it splits off, and then you have that population grows and then it splits off again. And anytime you do one of those founder events, you're taking with you just a subset of the existing genetic diversity that exists in the founder population. So, or in the parent population, I should say. So what you end up with is much stronger genetic drift, which means rare variants get lost much faster. So that's why you tend to see younger ages
get lost faster in small populations. You get a stronger drift.
Yeah, you get stronger drift in small populations, so you lose rare variants faster.
I never thought of it that way.
So you could model. You look at population sizes, and you could just model this you could just Google very basic kind of population simulators, and you could set the population size, the number of generations, and certain alleles at whatever frequency. And if you have a small population and a rare allele, all it takes is just you flip a coin, and it comes up tails just one, then that allele is gone, right? So when you have, and the origin of these single nucleotide variants are just mutations within these populations. So they are, when they first appear, they're necessarily extremely rare, right? If you have a population of 10,000 and one person gets a specific mutation, well, the frequency of that mutation is now 1 in 20,000, right? Because everyone's diploid. So it's 1 in 20,000. It's really easy in a small population to lose something that's at that frequency just by drift, just random chance.
Yeah, because what I do is I just imagine a hypothetical population of 2 people. I know that's artificial, and that's exactly what happens. Things will drift out faster in the smaller the pop. Okay, thank you.
But that doesn't mean selection isn't operating. It just means, relatively speaking, the smaller the population is, the more important drift is.
Yeah, this is actually why it's good to go through this, because then it refreshes my memory of how this works. Because there are lots of things in neutral theory and nearly neutral theory that I have to keep refreshing, because there's stuff that's counterintuitive. What was it? Mutation rate equals fixation rate, something like that. Just if it's neutral.
Approximately, it gets funky. Another approximation is frequency equals probability of fixation, right?
Yeah.
To a first approximation.
This is where the mutation rate equals fixation rate was really counterintuitive until I really had to think about it. Anyway.
You got to think about it. But then you do the math. You're like, oh yeah, okay, that makes sense. But you gotta work through it. Okay, so you go into these other classes, right? So nonsense mutations. This has a very specific meaning. A nonsense mutation is when you have a mutation within a coding sequence that changes whatever the codon is anywhere in that sequence to a stop codon. So this is going to cause a premature stop to protein synthesis, to translation at a ribosome. So nonsense mutations are definitely going to mess with protein function unless they occur at the very end of the sequence, right? But if a nonsense mutation occurs 50% of the way through a gene, then you synthesize half the protein and then stop. That's not going to work.
I'm embarrassed to say I actually didn't know that.
It's a weird word. It's a weird word for it, but yeah, that's what that is.
Okay, so there's nonsense, and then there's, yeah, the non-synonymous and synonymous. I get that. I've heard the term missense.
Yeah, so missense mutations are when you substitute one amino acid for another. That means non-synonymous. So you could say nonsense and you could say missense, but you more frequently hear non-synonymous. Okay, so
that's all right. Pun intended. Non-synonymous is synonymous with missense.
With yes, exactly. Yes.
Okay. All right.
Yep.
Thoroughly confused the viewers now. Okay, thank you.
So as you can see, those are more frequent, right? Non-synonymous, and they actually talk about in the text of the paper how they actually break this down further into different classes where, like, do the amino acids have the same properties or do they have different properties? And the more dissimilar the properties are, the rarer that variant is. So like if you take a basic amino acid and you change it to an acidic amino acid, or you take a very small non-polar amino acid and you change it to a very large polar amino acid, right? If you change the properties radically, then that non-synonymous mutation is less frequent, which is to say correlates with younger, than the ones that are where you take, one small non-polar amino acid and replace it with another small non-polar amino acid. So basically, the larger the effect is, you see this same type of thing where the larger the effect, the more recent the average age is when you kind of dig into this category a little bit more. And that makes sense because the bigger the change, the more likely you are to disrupt protein function, the more likely you are to be selected against.
Is that what they mean when they say purifying selection, that it's going to purify?
Exactly. Okay. Yeah, so you have variants that appear through mutation, but then some of them are going to cause a problem. They're going to be selected out. So that's purifying selection. And then synonymous, just it's a codon, CCC changes to CCT. They both code for proline. It maybe affects the translation rate a little bit, maybe. But other than that, it's not going to have a discernible effect. And then obviously non-coding, you're talking about sequences that are not within the exons at all, and those are well tolerated.
Well, the synonymous ones, I mean, this is a nerdy, the first class I took was on non-coding RNAs. They can be subject to the mRNA, the microRNAs.
Mm-hmm.
It can target that.
Yes, so you can have different codons. So your RNA sequence is going to be different, and then that's going to differ in susceptibility to interference with like microRNAs and stuff. Yeah, that can have an effect. So generally small, but yeah.
So then there's the non-coding. That's outside of the exome, though, isn't it?
Mm-hmm. Yeah.
So they just threw this in.
Yeah, so that's like a kind of your control region. Like, so you can think of the non-coding, unless it's like a sequence-specific regulatory region, then you can think of the non-coding is going to be not sequence constrained, right? So like this is the baseline where selection is not affecting these at all. And then the average ages are, in the 45 to 60,000 year range for those variants. And then as you get into the more and more constrained, you're going to see selection playing a larger role in getting rid of these, so they end up being more recent.
All right, so thank you for explaining that was used as a control group. So even though they were studying this study,
those are my words.
Okay.
I do want to be clear. Those are my words, not the author's words, but that's my interpretation.
Okay. Thank you.
So I think that's what we're seeing here. You're seeing differences in the persistence of mutations based on the strength of selection against them. The weaker the selection, the longer they're able to persist. This is not to say that there were no splice site mutants from 50,000 years ago, but a splice site mutant that happened 50,000 years ago has likely been selected out of the population by 30,000 years ago. So you don't see those anymore. But the ones that occurred 10,000 years ago, you still see them. Does that make sense?
Yes, yes. Now this is bringing back memories because I did study a splice site variant, lambda interferon 3,4, and I did want to tell the young Earth creationists, you got to be careful about the shared errors because I see them. I saw that same pseudogene in other primates, and sometimes if you reactivate. I don't know if they'd actually classify that as a suit. I think it was a situation where when the splice site was compromised, it would code one exon or something like that.
Yeah, because if you, right,
Yeah.
so who's not familiar with this, just background. So within a gene, right, you make your gene sequence. Well, most of that isn't, in eukaryotes, most of that isn't actually making, it doesn't have the codons to direct to make the amino acids for the protein. Only regions called exons have those. And then in between the exons, you've got introns, which are typically bigger by a lot, that don't have that information. So you go exon, intron, you kind of skip your way down the sequence. Well, you have to cut out those introns. And the borders, right at those borders, they have very specific sites that basically say cut here. And those sites are actually in the intron, right? Because they can't be the exon, because the exon's the amino acid coding part. So they have to be on the intron side of the cut, but it says basically cut right here. And if you mess up your cut by even just a couple of bases, it screws up your whole sequence. So you can't mess up the cut site. So if you mutate those sites to where either they cut in the wrong place or they don't cut at all, you've completely wrecked your amino acid sequence, and your protein is just not going to work.
Right. And so this splice site, I found it in, it's one of the examples of shared errors, which is very strong, would be very strong evidence to support universal common ancestry. And I've tried to tell creationists, you know, this is tough, guys. You need to explain this.
You need to have an answer for that. Yeah.
Yeah, that is, and that's why I brought up some memories too, because I could see that pseudogene in primates where the splice site was compromised. And then the reason it was an interesting case is some humans recovered a splice site and they had more immunity to colds. Well,
cool.
It's interferon lambda. They were able to express, yeah.
That's cool.
So
it's a pseudogene, but if you recover the splice site, then it can be activated in a functional version, right. And then that's pretty cool. All right,
okay,
so let's see.
This is a nerd session, guys. So every now and then we're going to go down some rabbit trails.
Oh, I like it.
Okay.
All right, let's go to figure 2 now. So this is looking at the. All right, so this is the average age. All right, so I have to remember what this is here. Okay, so we've got average age of non-synonymous and other Snvs as a function of the number of methods. Okay, so this is based on different ways of predicting that a specific mutation is deleterious. And remember, when we're talking about evolution, deleterious means harmful. It doesn't mean deletion. It doesn't mean you've cut something out. It just means, is it harmful in some way? So what we're looking at here is we're using a number of different models to evaluate the degree to which these variants are harmful. And then we're saying we're grouping them. So there were 6 models that were used to evaluate the whether they were deleterious or not, right? And we're saying based on how many methods from 0 to 6, where's my cursor? Can you see my cursor? I want you, there it is. Okay, so based on how many methods 0 to 6 found the specific variant is harmful, what's the average age for that variant? So what you see is that the ones where none of the 6 methods found that it was harmful. They're pretty old, 40 to 60,000 years. But as you assess them as deleterious in some way through some method, the ages get younger and younger and younger. And the youngest we see are 5 and 6 are pretty close to each other. I don't know if those error bars are going to overlap, but they're pretty close. And those are, within 10,000 years. And it's the same kind of thing as above in terms of the type of mutation, except this one's specifically looking at the effect of the mutation. So if it's more likely to be harmful, it's also more likely to be recent. And the reason for that is that, well, if it's harmful, then it's going to be selected against. So that's not to say there weren't harmful mutations that occurred 50,000 years ago. It's just that by the time you get to the present, they've all been selected out of the population, whereas the ones that happened 5,000 years ago are still here. So that's what this is showing.
Right. If I could just time out here, because this scenario can happen nicely in an expanding population for a variety of reasons. And if I could think out loud a little bit, because this will help me comprehend what's going on. When you have in a lot of population models, you have well stirring, but actually in a real population that's expanding and migrating, they're kind of isolated. So it's possible you could have kind of all these multiple founder populations where you have the mother and father, they have a new mutation and it's bad. It's not killing the kids and they're not competing, if their kids are competing with each other, it's like, well, they both have, assuming that they have it's possible that in an expanding scenario, they're not getting killed off because they're just multiplying. It's when they can actually compete in a well-stirred population, and this is over time. This is how an otherwise deleterious mutation can persist for quite a long time.
Yeah.
I mean, it's not instantly removed from the pot, especially in that scenario. Okay. All right. I just had to kind of.
Yeah. And remember,
that's the thing that kills me. Is each population genetic equation applies to certain models of the population structure, and it just drives me crazy which equation applies to. Okay.
We have to keep in mind that we're taking these 2 giant chunks that are themselves made of lineages that are not necessarily intermixing with each other, right? So you don't have one giant well-mixed population migrating into Europe, just like you don't have one giant well-mixed population in Africa, right? There's a number of subpopulations within each of those groups. So looking at this as, Europe and African data sets, we're kind of lumping all that stuff together into 2 pools when each pool has its own structure. And it's nice because we have so many Snvs to look at. So you're going to see some that are going to be younger or older just based on the population structure like you just described. But on average, you can look more at the aggregate level and get a trend. And that's kind of the big finding here by using I think it was like 1 point something million Snvs total were surveyed in here. That's a large number. That's nice. I like a data set of 1.1 million.
Okay, because I'm Asian.
That's good.
Because I'm Asian, I guess I could say this. I had to Google just to make sure. It's been kind of my perception that Asians tend to have more myopia incidents. There is a study, ethnic differences in prevalence of myopia.
Really?
And the highest levels in East Asia.
Really? Interesting.
Yeah. Okay. I didn't mean to go into stereotypes, but I said, okay, since I'm Asian, maybe I can get away with it because I've noticed Asians often have to wear glasses to correct for myopia. And someone did a study. So what we were talking about, the mixing of the populations, you can see that, maybe a founder population for the East Asians, they had myopia problems, but they weren't competing with other groups.
Right.
Yeah.
Yeah, you have in a exponential growth situation with low competition, you have relaxed selection generally, until you run up to that carrying capacity, and then you reimpose selection. But during that exponential phase, you're just going to generate a lot of variance, right?
Okay. All right.
Okay.
So I'm just trying to process what we are seeing here, how
Yeah.
it could.
So
All right.
another aspect to this is right up here, which is really cool, these little pie graphs, because here we have the averages, right? So like you've got, I mean, these are showing the same thing, basically. So this is for non-synonymous, and this is for everything else. It's the same trend. They don't show the full 6 here, but it's the same trend. But up here is really interesting, because if it's a black part of the pie, then that is showing that it originated less than 5,000 years ago. And the white part is 5,000 years ago or more. And so the really cool thing is you can see a trend here, right? Not just is the average older as you have fewer pings for being deleterious, but the frequency, like the actual fraction that's earlier than 5,000, later than 5,000, is very different, right? So if you look here, it's like, what is it, like a sixth right there for that one? That's older than 5,000. Over here, it's a teensy little sliver. Most of the deleterious fraction there is very young, right? Less than 5,000, which makes sense because the average is probably right around 5,000 there. So that makes sense, right? So these pie charts are just a different way of showing the same trend using 5,000 years as a cutoff point. The kind of the interesting thing here for me, and so, you know, having brought this paper to my attention, is like, okay, you get a lot of things that originated recently, but you also get a lot of things that didn't, right? That's the kind of the big thing here. And like, okay, yeah, that's within 5,000 years. That's a young Earth timeline. This is not, right? That is not a young Earth timeline. So I just want to point that out right there. That
Oh yes. And so the young Earth creationist there, that's something we need to deal with. And in the interest of, you know, this is why it's good to have you here, because I want my fellow creationists to hear what you have to say. If I could just time out a little bit here, you know, sometimes when the creationists are making their presentations, you know, they abbreviate it, and it won't have the other side. And this is kind of, that's why I was grateful you're coming, because then I could feel like I've given proper platform for the other side to air their viewpoint, and it's very important. So thank you.
I appreciate you doing that. You don't see young Earth creationists doing this. Of like, okay, we have data here that points to mutations that existed 50,000 years ago. Obviously, it's a problem. You don't see young Earth creationists grappling with this. So I really appreciate you doing this.
So just wanted to express thanks, Dr. Dan.
Well, thank you.
I
appreciate that. All right. And then this graph, it's functional, single nucleotide variants, percentages 0 to 20, and then the age, and it's the same thing. Basically, it's there's fewer that are older, which is exactly what you'd expect. There's not like too much groundbreaking there. Now figure 3, when I was reading this, I kind of skipped over. This is the kind of population modeling stuff. So Sal, this is actually probably more in your wheelhouse than mine, because this is looking at, this is looking at chromosomes. We talked about autosomes, right? So this is chromosome 1 through 22, right? Europe and European American and African American populations, and basically it's looking at pre and post population expansion, which would have begun, you know, I forget, the authors gave a number and I forget what it was. Actually, I'm going to scroll up because I highlighted it, I think. So I just want to find the number for where there's like, this is where the population growth started. So just give me 1 second here.
I didn't know you could highlight. I didn't know you could highlight your PDF.
Oh yeah, you just up here. Yeah, you get this. You get this right here. You just highlight it. I even use different colors. So that
I'm embarrassed to say I still have my old highlighters somewhere, the old ink highlighters from like
Here we
long
go.
time
So
ago.
so they're saying the population expansion began 5,000 years ago. And that's, oh, undo that. I just messed up my highlighting. There we go. So they're saying the population expansion began about 5,000 years ago. And that's important because what did we just talk about, Sal, with the growth facilitating the appearance of deleterious alleles that are more likely to persist while you're in that relaxed selection? Well, look at all those deleterious alleles that are younger than 5,000 years. That actually, like, that makes sense. So you come down here and you can just see where things are enriched in the variants, like where in the chromosome, which, like, okay. That's, okay. So you get more after the expansion than before, which is what we'd expect. That's what, so there are fewer here, there's more over here, fewer here, more over here. That's what we'd expect. There's nothing like groundbreaking there. And then let's see what's going on down here. Number of genes with functional Snvs. So this is how many genes actually have these variants that are functional. So they're less likely to be selected against, right? So you've got a few that are greater than 5,000. I think there's supposed to be a teensy little bit of pink here, maybe. Number of genes with functional, SNVs times 1,000, SNVs times 1,000. Okay, what am I looking at here? B. The bar plot summarizes the number of genes segregating one or more. Oh, one. And then this is more than one? Oh, no, I'm wrong. This is before and then after. Oh, and that makes perfect sense. Yeah, so this is pre-population expansion, and this is post. And the idea here is you get way more post-population expansion, right? So the less than 5,000, that's your pink bar right there, whereas these were pre-existing. So that's the overlap. The green is the overlap between the two sets, the old and the new. The blue down here is just the old, and then the pink right here is just the new. And that makes sense, that you get this explosion of diversity correlating with the timing of the population expansion. So that makes sense. And then there's one more figure, which, if I recall, oh, this is the Mendelian and the complex stuff. This is cool. This one's confusing. How are we doing? How's everybody doing? Sal, this is fun. I haven't done this in way too long. I'm just checking the chat, making sure everyone's good. Sal, how are we doing?
I'm doing great.
Sal here? There's Sal. Okay, cool. All right, so this figure, let's see. Okay, so we're looking at different types of, instead of looking at like the type of mutation, so synonymous, non-synonymous, missense, nonsense, whatever, here we're looking at the type of trait. So Mendelian essentials, kind of like you need that. Complex is going to be something that's like a polygenic inheritance. There's multiple genes affecting a trait, and then others, other. And what we see here is, let's see, this is going to be for, so this is the same thing we've shown before, that it most deleterious variants are recent. It's consistent across all these different groups. That's fine. And then we come down here, we actually break it out by functionality in the genome, kind of broadly speaking, right? Cellular processes, genetic, metabolism, whatever. And that's going to be average ages and all across 6 broad classes. Okay, so what they're showing is that the average age for deleterious variants is much smaller than the average age for everybody. So if you're harmful, you're much more likely to be recent. This is just another way to break out those same data. It's breaking it out by function within the genome. And a really interesting thing here is metabolism makes up a huge portion of this. There's just a lot of known variants, I guess, in metabolic genes, because they're fairly well documented. We've been studying metabolic genes since like the birth of human genetics. So I guess that makes sense.
Dr. Dan, if we could time out a little bit, just to
Let's
pause?
do it.
So the term for deleterious there is not that we've actually counted the effect of like relative fitness in terms of reproductive success.
Right.
In this paper, they're talking about you talked about the 6 methods that they evaluated.
Yeah.
So I would presume that they're really referring to kind of the medical effect, where it's they can measure some degree of severity, like it's disease association of some sort.
Let's see. We are going to find out. Oh, hang on. Deleterious. Okay, let's find out exactly how they define deleterious.
Maybe DAF stands for disease associated frequency.
Oh, maybe. Yeah, maybe. Let's see. Towards putative. Okay, so they used functional prediction methods. So SIFT, polyphen, likelihood ratio test, mutation tester, and then 2 conservation-based methods. Okay, so I can't comment on the functional methods here. So this is on page 3, 1, 2. It's the 3rd paragraph down. And it starts with, to more directly identify putative deleterious SNVs, we used 4 functional prediction methods. They named the 4, applicable to non-synonymous SNVs, and 2 conservation-based methods, applicable to all SNVs. So I don't know what the functional prediction methods are, but the conservation method, so the idea there is you're looking at the degree to which a specific thing is conserved in the genome, and you are saying, okay, if it is well conserved, that implies that there is purifying selection operating on that site. So variation from that implies that it's harmful. Does that make sense?
It does, but I have to throw out a data point here with Michael Behe. He had a forgotten experiment that is still being discussed. It's been forgotten, I'm sorry, that is rarely discussed. It was on mutation, it was histone deletion mutants, where we had 100% conservation on well, histones are very conserved. And I have to go revisit that. Histones. And they could not, he was commenting on an experiment someone else did. And there's no detectable change in reproductive fitness, which was surprising. And that experiment has happened in other cases. So I just have to throw that out there for the young Earth fans.
Oh, wow. That was from 1990. Let's see.
Histone deletion mutants. I think the experiment has been, now it's very interesting what this means, because Brenda Andrews did an experiment where she did gene knockouts, and she was actually surprised at the robustness of the genome. You could knock out one gene. You didn't detect it till you knocked out the second.
Yeah, you get a lot of redundancy. Yeah.
And so well, thank you for looking that up, because I was wondering if they had any medical evidence of functional change because of the robustness. Okay.
Yeah, and so the nice thing here is that you're using 6 different techniques to do this.
Okay.
And that's good, because no one technique is going to be perfect for this.
Right.
But if something pops on all 6, then you're like, that might cause a problem, right? And they broke out their data by the number of techniques that detected it as deleterious. So I think that's a useful approach. And that's,
if I may just say, this is where there's an open, I will just assert here, I think there is room for saying it may be expected according to the prevailing theory about purifying selection, but we may not actually know the effect, the degree that real selection is in play. It
was not directly measured. Yeah.
Right. There is a little bit of an inference that if it's conserved, there must be selection on it. I would agree to the extent that if you get large chunks of, like you take a protein and you knock out maybe one conserved site, you might get lucky. It doesn't have an immediate, even if it is, non-synonymous, even if you get, but it's when you get a whole bunch, and we don't know the degree of these that can take.
Don't know the degree of redundancy.
Yeah. So I would agree with you that the prevailing theory would argue exactly you did, what you said. This is an expected, that you would get purifying selection. And the creationists would say, we don't know that for sure. We would tend to we prefer neutral models, not just because it favors young Earth, but the calculations are easier to run through a computer.
So I will say these methods are not based on nothing. Like, these are methods that you're taking what you know based on actual data, right? And then you're applying those parameters to situations where you don't have the direct data, right? So we're not doing the biochemical testing to see if there's a direct effect for these variants. Because, like, it's humans, you can't just do like comparative fitness tests on humans, right? You can't do that. But what you can do is we could say, okay, we can look at yeast, we can look at mice, we can look at XYZ in the lab. And we can say, okay, if you meet these criteria and you perturb it some way, that tends to have a fitness effect of this magnitude, right? And we could directly determine that for other organisms in a laboratory setting. And then we could build these programs that say, okay, let's look for those same sets of parameters in other cases where we can't directly assess it.
And this is I'm just throwing that out there that, this, what you have stated would be the default position at this point. And what the young Earth creationists have to do is say that the selection might be weaker here. And this is why it's important they hear what you had to say, because this is not as, and I will admit now, I'm glad that we went through the paper. I was thinking about this today. I wouldn't rate this as a great evidence of young life, because for the reasons I just stated, we would have to actually demonstrate selection is much weaker,
right,
than the prevailing theory. And that's actually really hard to do. That's why I hate population genetics. Because in the population genetics model, it's like, how do we arrive at W, the relative fitness? And just like, population genetics models, we just assume it has a figure. We don't actually state how we arrive, either measure it or predict what it's going to be. So, but anyway, I hope I never thought we'd have a discussion on Behe's old 1990 paper on histone deletion mutants.
I'll tell you, I just looked it up, and the interesting thing about that is it's about molecular clock hypotheses. And like, dude, 1990 was, wild time, right? Just kind of the 80s, like, it was a wild 1990s, the Wild West for this kind of stuff. But like, you do not use sequence constrained regions to do a molecular clock. The point is you don't use regions that have purifying selection. You don't use regions that have recombined. You only use regions that are unconstrained to do a molecular clock analysis. It's like, but remember, 1990, we didn't even have a human genome sequence. We had nothing in 1990, right? So it's a lot harder.
Well, there are still some interesting results to that. And so I'm just going to throw this on the table. We'll go back to the paper shortly. That they're thinking it would be instantly lethal because it's 100% conserved.
Yeah, there's some of those.
And it wasn't. And there were some things in non-coding regions also more recently where they had these large non-coding regions in mice that were deeply conserved. They thought if they knocked it out, the mice would die, and they lived.
They didn't.
Yeah,
like, that's weird. Yeah. There's some weird stuff.
So the problem is, why is it conserved so deeply, and why is it that? And I remember I posed that to the evolution types. They said that is an interesting problem. How can it be conserved, and yet you can knock it out? And it was through two lineages that supposedly separated. So all this to say that I think we should be aware of the prevailing theory, but I will say the difficulty of measuring the effect of selection is it's really hard.
And in particular, it's tricky on humans because obviously we can't experiment on humans, right? You can't do that. That's unethical. You can't do that. So you can't compare. The thing that I want to point out here, it's actually perfect timing that you pulled that back up. The thing I want to point out here that's important to note is these are not points. These are not data points. These are data ranges, right? So the stuff you're talking about, right? You can look at stuff that's highly conserved, and in some small cases, you can knock out something that's very tightly conserved, and it doesn't have a detectable effect. But more often, if you knock out something that's tightly conserved, that's going to cause a problem. So what we do, which I love big numbers for data sets up here. Here we go. Yeah, 1.1 million. Yeah, it's nice. That's really good because you know what's great about a data set that's over 1 million is you swamp the outliers, right? You're going to get a trend. If there's a trend to be found, you're going to find it when your data set has 1 million points in it. So that's really nice because now we can look at this and it's possible. These error bars show the standard deviations. It's possible there's something in one of these two right here that's 40,000 years old, right? It's possible. That's going to be a big old outlier, right? Or maybe it's down here. Maybe it's super conserved, right? It's in here, right? And all those conservation-based methods were like, this thing is conserved. It's definitely deleterious if you mess with it. But it's one of those weird outliers, so it's actually like 30 or 40,000 years old. Well, that can happen, but on net, it doesn't, right? So you get this nice big data set, and you can account for stuff like that.
Can you go back to the, I think it was figure 1, panel A?
Yes, right here.
I want to, okay. I just want to read that. So DAF, disease-associated frequencies?
I don't think so. I don't know what that stands for.
That's really annoying me now.
All right.
Okay, I'll tell you why I'm asking. Just proportion to singular. If we had these created alleles, that is the rage in creationism, would it be on that right side of the graph?
Just 1 second. Just looking to see. Yeah. No, I'm trying to see if there's a phrase that has frequency in it, but it doesn't. Okay. Okay, so if you had created alleles, all right, you're saying you had created alleles 5,000 years ago-ish, 6,000 years ago-ish, right? Those alleles, well, so this is where you get pushed and pulled in a lot of different directions, right? Because if you had specific alleles, right, that are high functional, optimal functionality, whatever, 6,000 years ago, would they be over here? So the frequency is going to be very high in current populations. Maybe, but also you have to consider, well, you're talking about single nucleotide variants, because I'm talking about recombination then. So you have to have super high like warp speed recombination rates in order to get the variants you have. Even if you have created heterozygosity, you need to like accelerate your recombination rates by a factor of like 17,000 or something in order to get enough variants for the alleles that have a lot of, for the genes that have a lot of different alleles. But then would you still end up with like the created variants that are the most high frequency? I don't know. Well, the created heterozygosity, according to Jeanson, 99% of variants were created, and almost none have appeared through mutation. It's all recombination. So that would imply that what you should see in that case, because we're talking about single bases here, right? So what we should see in that case is a giant spike here with a long tail going this way is what you would expect specifically under Jeanson's model, because he's not accounting for any mutation. So you don't have this accumulation of rare variants. Instead, you start with everybody over here, and then you have a little bit of mutation. So you're going to end up with this long tail, but most variants are going to remain super common, and those are going to be the created variants.
I'm going to have to just chew on that. And so sorry, I just threw that out there.
No, it's all good. Yeah.
Okay. So now let me tell you what I was really interested in in the paper, and it was in the reference section, actually.
All right,
and that's what we talked about. I think it was reference 8. Yeah, that's the one where we got the that funny phrase where it said, because this is where I was interested. I said, how do they calculate how old it was? And that's been an interesting question, obviously.
That's the math.
That's where it says, using the overlapping genotypes of the Sanger, ENCODE, and SNP chip HapMap projects.
Okay, I can tell you what those are.
Dirichlet mixture. I'm just like, okay, that's
okay. I can tell you what those are. So, you said Sanger. What was the phrase? It was Sanger.
It's Sanger ENCODE.
ENCODE. Well, you know what ENCODE. Well, you know what ENCODE is.
Yeah.
Okay, so you got ENCODE. You've got Sanger, which there's Sanger sequencing, but there's also like a Sanger database or something. So these are all different sequencing databases. So what you're doing,
yeah. Okay, so I know Sanger. All right.
So what you're doing is you're looking at overlapping sequences in these different data sets, and then based on the origins of the sequences in the data sets, you can correlate that with population age. So if you have a sequence, for example, that's only present in like Morocco and the Canary Islands, then you could say, okay, well, we know when the Canary Islands were first colonized, so we can bracket when that sequence started to exist, right? Because we can know when the Canary Islands were colonized, and we know when the population that colonized the Canary or settled, whatever, right, from North Africa. So we can bracket like when that mutation occurred in the past. If you have a mutation that's present in, you know, European American population, I don't know if they went more specific, like regionally, you know, Irish, French, whatever, right, compared to an African American population from Ghana, from Nigeria, from South Africa, right, somewhere, wherever, you can use what we know about the human population structure and the ages of the different groups to bracket how long that mutation has been present within the population.
That's interesting. Okay, so this has actually been, you know, my main stuff on population genetics was Fisher's theorem, obviously, and things related. So I got very specialized in that.
We still have to do part 2 on that, by the way.
Oh, okay, great. We
got a whole chapter.
Goodness. I did have a lot of fun, and I still, you know.
It was fun. It was fun.
I actually presented to the Discovery Institute on that this past Saturday. But anyway, so the discussion on that paper continues. So in any case, I've always been wondering, how do they, and there's the word coalescence. I saw the word coalescence, and I studied it. Can you explain to me and the audience and refresh my memory and maybe clear up my my understanding or clear up my misunderstanding? What is coalescence? How does that relate to most recent common ancestor?
Okay. So coalescent theory is the idea that you have a lot of existing genetic variation, and you can look at things like the frequency of specific mutations. You can look at things like the substitution rate. That's the rate at which new variants fix within the population. And so you can look at all these different existing sequences, right? And you can work backwards based on the rate of accumulation, the per year rate of accumulation. And you work backwards to a hypothetical common ancestor from which all that existing variation is descended. And the idea here is you're looking not just at the number of differences, but what those specific differences are.
Oh, Dr. Dan, you muted yourself.
Okay, hang on. Am I back? Can you hear me?
Yeah, it must have.
Oh, no, I can't hear you. Uh-oh, hang on. Hang on a second. Oh, no. Okay.
Right when you're getting to right when Dr. Dan is getting to the interesting part.
Hang on. 1 second. Sal, can you hear me?
I can hear you. Can you hear me? Can the audience hear me? Audience, can you hear Dr. Dan when he's
I
speaking?
don't want to do that.
How many people?
How are we doing now?
I can hear you.
I got nothing.
Did you run out of battery power?
Uh-oh.
No. Right when we got to the interesting part. So I'm typing this so Dr. Dan can see. Matt has to hit the sack. Good seeing you. And I'm going to send a message to Matt. Oh no, not when we got to the most interesting part, coalescence theory.
Can anybody hear me? In the chat, if you can hear me, tell me. Let's see. Oh, one cow can hear us both. But Sally, you can hear me. Okay, great. Okay, so why can't I hear you? What's going on here?
Can you guys
Sal,
hear
can
me?
you say something?
Yeah, I'm saying something.
Yeah, I can't hear you.
Can you guys in the side chat tell me if you can hear me? Can you hear me, guys?
So why can't I hear Sal?
Did I? Can
anybody hear Sal?
Me? So I'm talking and I'm asking. I just typed in the chat, asked them to respond.
We're doing so well. Everything was working.
Right when we were talking about coalescence.
Like too many things open here. All right, we're going to close that. Maybe that'll help. If this is still broadcasting, it's going to be really funny that you could just hear us like see our faces trying to talk. All right, I'm going to talk. If you can hear both of you, but I can't hear Sal. I'm going to change my. Hang on one second here. Let's change that, and now let's go back to that. So Sal, everybody can hear both of us, but I can't hear you.
That's right. I can hear you.
You're back. We're good. Okay. All right. I don't know what just happened. Everybody.
Okay, you can hear me now. Okay.
All right. Let's do it. Okay. Coalescence, and then
coalescence and MRCA. This is what I was waiting for.
Okay. Oh man. So coalescence, the idea that you can look at the present diversity and you can work backwards essentially to a common ancestor based on what you know about the frequency of mutations and the rate of fixation within the population in question. So you can look, for example, at all of humanity, right? And you can look at autosomal DNA, you can look at the mitochondrial DNA, you can look at Y chromosome, you look at whatever. And you can work backwards to a common ancestor, right? That's the idea with coalescent theory. And it involves math.
Okay, so you're, I mean, I'm just sorry to ask a pointed question. Have you ever done a coalescence analysis yourself, or did you use software?
I have not. So I am well versed in how it works. I have not done it myself. So my work in grad school was all viral phylogenetics. My specific work in the lab was I was doing things like looking at the spectrum of mutations and how asymmetries in mutation rates showed up in existing sequences. So like I was doing the work under the hood that would inform the nucleotide substitution models that my colleagues were using to actually do the phylogenetics for different groups of viruses. So I wasn't actually doing like the phylogenetics. I was working on the parameters that you would use to inform your model kind of upstream of actually doing the phylogenetics.
Okay, so this is why I couldn't understand Dr. Jeanson's stuff. I said, how do you have coalescence models and MRCA? And after 2 hours, it's like, I don't get it. And I'm like,
Wait, hang on. Wait, hang on. So you got to ask Jeanson how this all works?
I spent 2 hours. And so I'm just going to the audience here and said,
can you get him on? Can you do that?
I probably can't, especially now that I said that, but
because
he was very kind.
Because I got some questions for how he did his coalescent work. Okay, I got some questions for Dr. Jeanson.
So I was okay. So especially the mtDNA, I said, okay, there's heteroplasmy here. This is going to be a nightmare. And he was trying to walk me through it. And then I started going through the papers, and that's when I threw in the towel. I said, I want to go proteins because we don't deal with this.
I have some stuff here. Let's see.
All right.
Okay.
I know this is okay. So guys listening, I'm not saying he's wrong, but this is one reason I haven't defended his work. If I can't understand it, I'm just like, okay.
I'm saying it's wrong.
Yeah. If I can't understand it, I feel uncomfortable. So you've heard me defend Dr. DeWeese because I've worked with him and we don't deal with population genetics. It's the biochemistry. You've heard me defend Change Tan and Rob Stadler, their Stairway to Life. Dr. Jeanson, this is unfortunately where Monkey for Banana and I had he argues Dr. Jeanson. I'm just like, obviously I'm on his side and we've been persecuted together. But if I can't understand it, I don't want to defend someone's work if I don't understand it, except to say, well, this is what he says. That's about all I could do. But then my conscience would bother me and say, what did how do you respond to Dr. Dan? And it's like, well, I can't because I don't understand Dr. Jeanson's work. It's not my place. And I don't feel comfortable. And I've gotten burned before, thankfully, mostly before I became a professional in this, by endorsing stuff I didn't understand. And I said, I'm not going to do that. And we were talking about teaching young earth creation responsibly. This is an example. It's like, okay, my conscience would bother me to say Jeanson's right. And I really don't know that he's right, especially given my position in the creationist community. Okay. So just suffice to say, that's why I was interested in understanding the coalescence math. Of course, Joe Felsenstein for a while was glad to teach me stuff, but then when he started seeing how I was using it, he wasn't quite too happy with me. And so Dr. Felsenstein was very kind to teach me, and I think I tried his patience as far as I could. And it led a lot to the success of my paper. So guys, I love Nathaniel to death, but I'm just telling you this. The reason I'm also saying this is I've been trying to tell people, why don't you play with proteins? You can have a little easier argument. This is so hard because there are a lot of uncertainties. So I have to, hang on, I have to take care of, let's see, hide this user from the channel. Okay, so I dispensed with someone in the side chat.
Nice. So Sal, if you want some time, we can. So I've thought a lot about Jeanson's stuff. I've written a lot about Jeanson's stuff. We can go through it, and like I can make Jeanson's case. Like I can go through and be like, here's the data,
right?
Here's the numbers, here's the math you do, and here's what you get. And he had like 6,000 years or whatever for the mitochondrial most recent common ancestor. But then I can also show exactly why that's wrong for like a lot of reasons.
So, and I would be interested, and we can do that. And so that's why I asked you a little bit. I wanted to have access to the materials that can tell us how to do coalescence theory. And it's my understanding and this is why I'd be curious to find out, in an expanding population, is it easier to determine the MRCA? And that's,
I mean, it's different. So you have different population growth models, you know,
yeah.
exponential growth, consistent, like you have different growth models. I don't know if it's easier or harder. It's just, it's different.
So that, do you have colleagues that we could ping on questions like that? Because
I mean, the people that I specifically know about this all do viruses. It's a little bit different.
yeah, okay, all right. So this is
It's a little bit different.
okay. So this is exactly, you know, this is an example of, and Erica made a good point. You have all these creationists kind of out of their specialty, and
Right.
I felt very comfortable defending topoisomerase because my colleague has studied it for like 18 years, published in Nature. I consider that in his specialty. You don't see him on the creationist circuit because he's a professor of biochemistry. He doesn't have to be out there promoting creationism. And he talks within the specialty, which is enzymes and topoisomerase.
Which is why I do evolutionary genetics. That's what I do. This is
right.
This is what you're gonna
so,
this is what you're gonna see talking about.
So you know, Erica made a really good video today, by the way. It was painful to watch.
Was it the young earthers versus their guests? Was that the one?
Yeah.
That one was great. Yeah, that was hilarious. I enjoyed that a lot.
It's like, okay, you know, we talk about teaching YEC responsibly. I'd make every young earth creationist wanting to go to college. It's like, this is what you're up against. You're going to have colleagues like Erica. All right. You just got to be aware. And you sometimes on the internet, people will just want to just fight and obfuscate and just do all sorts of things. And it's like, you're not getting to the data, you know, and she showed that model. So this thing with the coalescence, it's like, it's been bothering me a long time. It's like, how do they do this? And I haven't seen it in Joe Felsenstein's book. So
we can definitely talk about it.
Yeah. Okay. Well, thank you very much for, and that was the part that I was really wanting to look into it. And then when I read parts of the paper, it said this has been done, and it had the references. It didn't actually describe how they did it in this paper.
Right. Yeah. So we go to there's a lot of good papers out there on human most recent common ancestor calculations, and they get into things like population growth models and the strength of purifying selection over time. And you have to account for that stuff. So there's a really good paper from 2009 that I can think of that goes into it. And I really like that particular paper because they calibrate it against known migration dates, so, like recent ones too. So they'll calibrate it against things like when specific Pacific islands were settled within the last 5,000 years, right? Where we know like very specifically. So you have these dates that are locked in time, and then you can check your model against those dates. And that's a really nice validation.
And I recall that's one of the criticisms he fielded against Jeanson's work was that that particular instance where we knew when they migrated.
Right. Yeah, because it's always, you know, assumed phylogenetic rates. Like, no. We have specific dates, and we can check our models against those. Yeah.
I'm going to give Rebecca a hard time. Yeah. Here.
Hi, Rebecca. Yeah, we're not going to be. It's actually, Sal. It is actually midnight, so I should probably go to bed like an hour ago.
Thank you very much for your generous time here. And
that was fun. That was, you know.
Rebecca, if you'd like to come on for just a little teeny weeny time, and you could replace Dr. Dan. Dr. Dan, thanks for joining us.
All right. Thanks, Sal. I appreciate it. Have a great night, everybody.
Yes, take care. Oh, one cow stampede. Oh, this is so generous of you. God bless you. So you're getting Rebecca, you're getting some class here, but I guess she hasn't heard me. I really want to acknowledge One Cow Stampede for the super sticker. It's very generous of you. Thank you, and God bless you for that. So this this has been an example of what it's like for a creationist to go to school. It's also an example of why I get flack from other creationists, because I'll frequently say the evolutionists have a very good argument here. And those of you who know me know when I will forcefully insist when a creationist argument is good. I have promoted the Stairway to Life as being very top rate, and I've never had any challenges to that. Whereas stuff that deals with the trying to infer how far back like Adam and Eve were, that's been very challenging. And even given that I have background in and even published in population genetics, that I don't know how to do coalescence, because most of my work has been on Fisher's fundamental theorem of natural selection. It is, it's difficult for me to give an endorsement to certain figures and saying this is defensible. I will just say that the people who asserted it, I'll leave it to them to defend their viewpoint. Whereas some of the stuff that Change Tan has done and Rob Stadler, I can put out that book. I reviewed it and I said I could defend it as is, because it comes straight out of almost straight out of college textbooks. In fact, Stairway to Life is very much like the molecular biology course that Change Tan teaches, and so it feels very defensible. It's very clear. Whereas stuff with population genetics and having published in it and studied it, it's like this is not easy, and there are too many uncertainties. So notwithstanding that, I'm sympathetic to the interpretation that what Jeff Tomkins passed on as far as this paper is promising evidence that our genomes are young, but the problem is we're not able to measure and ascertain for sure how strong the selection values are. And I'm also curious how they're able to infer that something existed 80,000 years ago. So this is going to be an interesting conversation. So while I'm here and I'm saying goodbye, and let me write Rebecca here. So Rebecca, are you there? Do you want to join the stream for a few minutes? Okay, so let's go through some of the questions. Does the heat argument have any merit? Absolutely. Absolutely it does. And young Earth creationists have been arguing for accelerated nuclear decay. There is a challenge here that I should have put forward. Young Earth creationists like the C14 argument, and I'm one of those. If you have accelerated nuclear decay, you might wipe out the C14 signatures. So that's why I'm reluctant to invoke what they call universal. The formal term is isotropic, accelerated nuclear decay. And it causes a heat problem. There's some ways to alleviate that if most of the isotopes are at the continental surface level, crustal level, and not through the entire Earth. And this raises the question of the origin of the Earth's radioactivity. So there are ways to alleviate it as far as the heat coming from radioactivity. If we don't have an accelerated nuclear decay model that's universal, what they call isotropic, and also if radioisotopes didn't exist until the time of the flood, that's part of the hydroplate theory, and there's evidence of that given that uranium is 70 times more abundant on the continental crust than it is on the seafloor, which indicates that perhaps radioisotopes don't go all the way into the Earth, and that would alleviate the heat problem. The other thing is, what's the mechanism of accelerated decay? So the hydroplate gives one mechanism that I like, and it would localize the acceleration and also deal with the heat simultaneously. That's in Brian Nichols' video. So that's one instance where it pays to listen to the evolutionists' criticism, and maybe they're right. Maybe God has sent them your way to teach you something, that you're going down the wrong path. And so this is what I've been saying, is that the next generation of creationists, and for some of you who joined late, we have people like Joe DeWeese, who started out as young Earth creationists, got their PhDs, and still stayed young Earth creationists. I'm concerned that how we equip people that are going to go into a hostile university environment, and how they're going to be able to manage that, because they may be confronted with questions that the creationists have represented as being airtight, and it's not. It might still be true, but it's not. It's something that's not defensible. So let me, Rebecca, this is going to be just too brutal, I think. You might want to watch this if you have insomnia. And don't forget to ask Dr. Dan about topoisomerase evolution. Rebecca is hosting Dr. Dan on Friday on her Bread of Life channel. So, okay, God, teach me to pray your word. I'm going to give you a video, God, teach me to pray your word, that will explain how the flood is connected to radiation. So let me dig that up. Origin of, oh, by the way, let me big thanks to Dr. Dan for coming here and spending 2 hours with me. Hang on, I'm going to get you that link, and I'm going to drop it in the side chat. Okay. Good question. Okay, let me see if One Cow Stampede has any questions. He was so generous. All right, I have to admit, most of the time I was here, because I was so busy trying to pay attention to what Dr. Dan was teaching me, I didn't have a chance to, I didn't really look at the side chat at all, just except to see if there were bots spamming us. So if you did have, any of you had questions, the 4 of you that are, 3 or 4 that are here, and you wanted to ask, now's a good time. So Benjamin commented, I actually think most variants are rare single nucleotide polymorphisms. Yes, that is correct. And so the outstanding question was, what you know, what are the variants as far as when we talk about alleles that are common, like say, you know, eye color or whatever, and there are various alleles that are well defined, like in terms of like, can you taste cilantro or not? So, okay. 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 All right. Well, maybe that'll do it. And I encourage you all to go to Logical Plausible Probables channel to celebrate. Thank you very much. And thank you to special thanks to all the patrons tonight. Thank you so much for your generosity. God bless you. And